I'm sure the scene above will be familiar to you. It's a row of cherry trees, all in bloom together. It was a scene very familiar to me when I was a teenager, out of my bedroom window. It's beautiful, it's spring, we all love spring, we all love the cherry trees. But have you ever asked yourself why they're all flowering at the same time? It's quite remarkable when you see the cherry blossom. All the trees will flower at about the same time. Now, are they flowering at the same time because they're producing some gas, maybe, talking to each other somehow? Or are they flowering because of perceiving the signals in the environment and then independently making that decision to undergo the transition to flowering? And it's the latter. Plants can't move. They have to thrive where they are. So they have to follow the environment. They have to monitor temperature, day length, many aspects of the environment to actually be able to thrive and produce seed. So plants use many environmental cues for day-to-day to monitor, to regulate their growth and metabolism, but they use seasonal cues to time their development. Now, there are many developmental transitions in plant development. The transition to flowering is what I'll talk about tonight. But there's also transitions to senescence in autumn, the transition to germination, transition to making seeds. And all of those use different seasonal cues to align their development with the environment and the seasons they find themselves in. Now, flowering is the one we know most about. We still know really remarkably little about how plants actually monitor these seasonal cues over many weeks. But it's clear that both temperature and photoperiod are very important for flowering. And for the gardeners in the audience, you think, well, in my garden, different plants flower at different times. They must be monitoring different aspects of the environment. And that's also true for many agricultural plants. Rice, for example, is the limiting in terms of how high the latitude it can be grown because it requires short days to flower. Winter wheat needs long days, but also needs to go through winter before it will flower. So what I'm going to do in the talk tonight is give you an overview of these seasonal cues that plants use to judge when to transition to their flowering stage. And then I'm going to dive into the bit of the flowering that I'm most passionate about, and that's this ability for plants to sense and remember they've had the whole of winter before they flower. Going back to our cherry trees, how is it that they're all flowering in just the right time in spring? Very, very similar time. How have they managed to go through the whole of winter, judging the temperature and the photoperiod, and then quite independently make the decision to flower at the same time as the neighbor tree? So second part of my talk will be sensing and remembering cold exposure and our understanding of the molecular basis. And then the last part will be thinking about adaptation to different winters. So of course, winter's very different depending on where you are in the world. Plants have had to adapt to very different winters. And it's really important that we understand that adaptation because, of course, as winters are rapidly changing, we have to understand, are our plants going to adapt fast enough? And what do we need to do to our agricultural crops to ensure that we have good food supply? So, as I said, my passion is overwintering. It sounds a bit strange as a passion, cold temperature, long periods of cold temperature. So how on earth did I get into being passionate about cold temperature? Well, I got passionate about plant biology by doing a practical at university. So this is me, aged 18, mouth pipetting isolated chloroplasts, which we'd never be allowed to do anymore. Very excited because I could extract the little green energy producers from the leaf cells, put them in what's called an oxygen electrode, and watch them fix carbon and emit oxygen. And you can play around and do all sorts of things. That's what really got me excited about plant biology. So I stayed and I did a PhD, much against my better nature at the time, but it was very fun time. And while I was there, my supervisor went on a sabbatical to UC Riverside in California and came back really excited about what a fantastic time she'd had. And so I thought, oh, well, I don't really necessarily want a career in research, but California sounds quite nice. Maybe I'll go and have a job in California for a couple of years. So I then went off and did a postdoctoral fellowship in California, actually at a company. At the time, it was the beginnings of the whole genetic modification of plants. And it was a company near San Francisco, Oakland, California. Now, that was a very formative time in my life. I learned a lot of science. I met a lot of colleagues. But I also got to explore California a bit. And really, what was striking was seasons in California were really very different from seasons in the north of England, where I grew up, and the University of York, where I'd been a student. Here you can see the Southern California desert bloom. There was a super bloom about 3 years ago when it had rained a lot, and so there was a lot more flowering than there usually is. And so, I began to think, gosh, yes, my cherry trees back in Manchester, they were sort of nice and familiar. And here it was so different. So how are the plants responding to these very different seasons? Wetness triggers it in the desert, cold in Manchester. And one event actually then really sealed my interest. And I had gone into a nursery garden to buy some tulip bulbs because I felt slightly homesick, and I thought a few tulips might make me feel a bit better. And the man said to me, now, before you plant them, put them in the fridge for 6 weeks. And I was absolutely amazed. I still remember to this day this being stunned that they actually need the cold. You imagine that, well, not much happens in the cold and they wait for the warm. But no, these plants actually needed the cold period. So that's what got me reading up about it, and that's what got me excited about working in this area. So when I had the opportunity to start my own lab, and this was at the John Innes Centre in Norwich, famous, of course, because of John Innes potting compost. Most gardeners have heard of the John Innes name. This is a big plant and microbial science centre in Norwich. This is probably about a 4-year-old picture. They offered me a position to have my own research group. So I said, yes, I'm going to figure out how plants monitor the whole of winter and use that information to accelerate their flowering. Now, that's a process called vernalisation. And at the Institute at the time, because we'd had absolutely no idea about the molecular basis of any of these processes, what we did know was some understanding about the genetic basis, because vernalisation is very important for many of our major crops. So oilseed rape, winter wheat, barleys are all bred both winter types and spring types to extend the geographical range where they can be grown. So winter types are sown in autumn, then they overwinter very slowly, and then flower in March, April, whereas the summer types spring types are sown in March and April, so they flower much later. So there's been a lot of genetics, a lot of analysis, but no molecular information. So the Institute was quite interested in having someone attack this problem molecularly. Now, the problem was vernalisation. We know the genetics, but we don't really know anything about the molecules that were involved. And that was a process that was quite difficult to study in crop plants. So about that time, which was sort of the late 80s, the international plant community decided they were going to have to tackle these very complex processes through a genetic approach linked with a molecular approach. Molecular genetics, it's called. And there aren't many organisms you can do that with. The one the plant community adopted was this little plant on the right called thale cress, a Rubbertopsis thaliana. Now, I've brought some Rubbertopsis with me so that you can have a good feel, squeeze, whatever. They're non-transgenic, so they won't hurt you. There's no chemicals, so you can squeeze and whatever. And if anyone wants to take them home, be my guest. I brought them all the way from Norwich, and I don't really want to take them all the way back on the train. So you will find them in your garden. Probably they've stopped flowering by now, but they are very common in this part of the world. So that's what we're going to be talking about, a Rubbertopsis thaliana. So in the late 80s, internationally, the plant community decided if all of the researchers focused on one organism, then we could really share pool results and things would go much faster. And it was an organism that had a tiny, tiny amount of DNA in each cell. So you could actually sequence the whole of the DNA quite easily. And that meant we could then take the genetics, and with the genetics you can actually break a gene. Let me see. Does everybody know what a gene is? Hands up if you've heard of a gene. Okay, just so I know I'm on the right wavelength here. So you can break the gene by mutation, and of course if you can then sequence, you can identify the effect of that mutation and actually link it to the gene that you've broken. So you can clone the gene based on the mutant phenotype if you have the whole sequence. And for Rubbertopsis, we managed to get the sequence quite quickly. It was actually before even the human sequence. So we could then go in, make new mutants in our flowering process and in our vernalisation, and then isolate the genes based on whatever gene we'd broken. So for the first probably 3 or 4 years, there were several groups internationally all working together saying, let's look at lots of aspects of flowering time. Some people studied photoperiodism, we studied vernalisation. And so we went in as a joint consortium internationally to say, let's make new mutations to identify genes required to promote flowering in response to temperature or long days or hormones, lots of things. And we got lots of them. You do that by treating the seedlings with chemicals or gamma rays, as shown on the slide here. Most of the plants would still flower normally. The 1 or 2 would flower late. You knew you would hit a gene required to promote flowering. So that was how we got floral promoters. We also, surprisingly, actually got proteins that delayed flowering, floral repressors. And we could then put all of those together, do lots of genetic analysis, and actually really understand the regulatory hierarchy that was important in Rubbertopsis for temperature, for day length, for all of these different influences, and how they fitted together. And that led to really quite a big step change in our thinking of flowering, and that was that there's one relatively simple regulatory hierarchy that can explain flowering in Rubbertopsis, but also accounts for flowering in all organisms, in all plants. Now, this is a very schematic version of it, but it in essence says that all of these inputs, whether it be the photoperiod or the hormones that are promoting flowering, target a set of genes required for the transition. But they all target the same set. So you can induce plant hormones or you can increase the temperature and the same genes will switch on. And interestingly, the repressors were also targeting the same genes. So now you could begin to imagine, and let me just add one thing, the winter was targeting the repressors. So the winter switches off the repressor, which switches off flowering, and 2 negatives lead to a positive. But anyway, this framework then actually explained this diversity of flowering responses that people saw in plants. You know, going back to your gardening, why does petunia need long days and chrysanthemum need short days and winterweed need overwintering? People thought there must be completely different molecular mechanisms to explain that physiological complexity. But no, actually, it's one fairly simple framework. It's just that the different inputs become predominantly different in different plant species. So by doing the genetics in Rubbertopsis, we then could actually have a framework to explore flowering time in a lot of other plant species. So that's been really very important. So my passion was overwintering, so when we got this framework, we then started to focus on, well, okay, how does winter promote flowering? What's this floral repressor that winter is inhibiting? Now, vernalisation is a very significant process for a plant, because if you need to vernalise, which basically means you need 4 or 6 weeks of cold to actually promote flowering, you overwinter, it means you only have 1 generation a year from a plant point of view. And these are what are called winter annuals. You put them in the ground in autumn, they overwinter and they flower in the spring, like the winter wheat. And the other type of strategy is a summer annual or a rapid cycling strategy. And those are very significant because actually in some climates, you can have multiple generations a year. So winter annuals, for example, are very common where there's a very, very dry summer, where in fact the plants would die in the summer. So they the plants adopt a winter annual habit because that's the most reproductively successful. And of course, evolution is always going to select for the most fittest approach. Whereas in England, summer annuals are very common because they can often fit a couple of generations in. Now, the joy was, in Rubbertopsis, we had these winter annual and rapid cycling types. Now, if you were an audience, I talked to, a sort of classic Rubbertopsis audience, they only ever see the plants on the right. People use a Rubbertopsis generally because it's rapid cycling. They can do their genetics really fast. You can go from seed to seed in about 6 weeks. So somewhere, the plants, if you can have a look at, some of them are flowering, the plants I passed around, and some are not. But actually, the majority of Rubbertopsis, if you go to somewhere here or further north, up to, say, Sweden, and put them in your nice, cut the seed and then put them in your nice, warm, long-day chamber, they would look like the plant on the left. They would, because you hadn't given them any cold, they keep making leaves. Whereas the plant on the left, well, I've got my left and rights, yes, if you give it 4 weeks of cold before you put it in your long-day chamber, it looks like the plant on the right. It fully fertilizes, it fully accelerates its flowering. And the one gene that is really important in this is a floral repressor. It's a protein that blocks the activation of all those genes required to make a flower. And it's predominant, it is a major effect in Rubbertopsis, as I'll show you. So you're going to hear a lot about this gene, FLC. Trendy little name called flowering locus C because it was named many years ago by somebody else. They weren't very interested in it, so they had flowering locus A, B, and C, and we've ended up being stuck with the name. But we've spent probably 25 years working on this gene. And when we look at these really, really late flowering winter annual types, it turns out that FLC needs to be activated or enhanced expression by a protein called Frigid ER. And again, this was named in 1955 by an old geneticist. And I put a picture here to just give you a sense of what's going on. If you take an analogy, FLC is like the brake in the car. And what Frigid ER does is apply the brake. And it's a very strong brake, such that you really need a lot of cold to switch off FLC. So this is why those really late flowering plants will not flower unless they have cold. So you've made an awful lot of the brake. But the striking thing was when we then looked, remember there's the winter annuals that overwinter and have one generation a year, but then there were these other type, the summer annual or rapid cycling type. They turned out to be a very simple change. They had lost the ability to make Frigid ER. So it was just disrupted. So in a single mutation, you can change very significantly the reproductive strategy. That was quite a surprise that it was as simple as that. Winter, on the other hand, doesn't go through Frigid ER. Winter affects FLC itself directly, and it shuts the gene off. Okay, so at that point, we had quite a good skeleton understanding of the regulatory hierarchy and a beginning of an understanding of the genes involved in this need for winter. So now I'm going to get a little bit more elaborate onto the molecular side of what is involved in the sensing and remembering the cold, which is suppressing now expression of this gene, FLC. Now, vernalisation has some interesting properties. If you give plants small amounts of cold, they partially vernalize, and if you give more and more cold, they vernalize more and more and more. So the flowering is accelerated progressively. And no one could quite understand how that might work at the molecular level. And the second really interesting thing was if you give a plant a cold period, and then you take a cutting, and you regenerate a brand new plant, that brand new plant will remember it's had cold. So all those cell divisions are remembered. Something the cold has done is stable through cell division. So when we actually began to look molecularly at FLC, and this is now a way of looking at the expression level of the gene, black just means there's lots of it, you can see that with no cold, there's a lot of it, and that's when a plant would be very vegetative and make lots and lots of leaves. But as you give increasing weeks of cold, then the gene very gradually switches off, and you get less and less and less of that gene being made. But interestingly, if you take, let's say you give them 2 weeks of cold, and you take it out and put it in your chamber, it will stay at that intermediate level through all of the rest of development. whereas if you've given it 4 weeks, it will go much lower. So at the end of the cold, wherever it's got to, it's then fixed at that level through the rest of development. And that's called an epigenetically stable process. It's a process that is responsive to something, in our case cold, but then is fixed at that level through cell division. Now, epigenetics we're going to hear a lot about, and can I have another arm raised so I know how much background? Who's heard of epigenetics? Okay, quite good, thank you. So we've got an epigenetic process here induced by cold. So if we look at the life cycle of a plant on a graph, so you start off as you germinate as a winter annual with very high levels in the autumn, and then slowly but surely, as you saw on the previous graph, the gene is switched off during the winter. It then remains at that very low level through the spring and the summer, so the brake is off, and now all of these positive factors, the photoperiod, the hormones, and all of these other factors that are required can actually do their job now and get these genes switched on for that transition to occur. In fact, for FLC, once the seed are being made, and what you see in that sort of funny little thing in the black box is a seed pod. So that at some point before the seed is fully formed, you have to switch the gene back on so that every generation, as they germinate again, need to go through winter. So we have an epigenetic switching mechanism here, switching off slowly and quantitatively in the cold, remaining off for a very long period as the plants are growing and flowering, and then switching back on again as the seed are being formed. And the mutants that we characterized were unable to hold that memory. So they often went down in the cold, but the gene was then switched back on again when the plants came back into spring. Now, by looking at these mutants and saying what are the genes that we've disrupted, we went straight into the heart of epigenetic regulation. And actually, this is now epigenetic regulation that is important in our own bodies, in all animals, in all plants, all higher organisms above the budding yeast will have this level of regulation that I'm about to describe to you. So epigenetic regulation, it's called the layer above the genes. I've got a schematic here where you've got the chromosome unfolding. So this is the chromosome at the bottom, unwrapping. Okay, so as it unwraps, when it's fully unwrapped, you'll see the double helix. But in that intermediate phase, you'll see that the DNA wraps around these balls, these purple balls, which I'll show you a structure of in a minute, are called nucleosomes. They are basically proteins which are very conserved proteins, and the DNA wraps around, and it helps concertina a lot of DNA into a small nucleus. This structure of this is called chromatin. So chromatin is the, if you like, the building block of your chromosome. And that chromatin is modified chemically. The proteins are modified, and also the DNA is modified. And so people have often used the analogy of a computer. So remember, every cell has the same DNA, but every cell does not do the same thing. So what are the instructions that tell this cell to do one thing and this cell to do another thing? So the DNA, you could imagine, or the gene, is the hardware, the DNA sequence, and then the instructions delivered are the software, and the software is then different in every cell. And the software are these chemical modifications that are delivered, which are then stable through cell division. So when they're delivered, they're then inherited. So even when the initial signal's gone away, they stay in that state. Now, as I said, this is very important. Epigenetic regulation underpins growth and development in most organisms. If you look at a cell and then all of the cells that derive from that, from an embryo cell, you have to give cells instructions in the embryo. Those instructions often then go away. But that cell remains in that state, a skin cell, a hair cell, whatever, through many, many cell divisions. They're epigenetically programmed. Twins, of course, have the same DNA, but have epigenetic differences, which is why a lot of diseases are epigenetics, and they've been studied in twin studies. And, there's a lot of information now that many cancers are the result of epigenetic changes rather than genetic. And the initial epigenetic changes then tend to cause more mutations in the DNA. So a lot of work is going on in epigenetics at the moment. You'll see a lot in the press. And it's the same in a plant, the same mechanism as it is in a human. So if we go back to our chromosome that you saw before and blow up one of those purple nucleosomes, they're very beautiful. They're a big structure on the right there with the proteins all color-coded in the center with the DNA wrapped around. I'm going to draw that schematically as this, okay? So that's the limit of my PowerPoint expertise, so I'm going to show you some of those, but that's a simple version of a nucleosome. And if you look at the nucleosomes and the chemical modifications, we won't get into what they are, but we'll color-code them as modifications that are conferring an active state. We'll color-code them green in a traffic light system. And modifications for a silencing state, we'll color-code them red. And when you look at the chromatin under a microscope, you can actually sort of see the beads, which are linked by the DNA, if you spread them out in a certain way. So when we then went and looked at our FLC gene during this cold, indeed, when the gene is on in autumn, the chemical modifications were green around the nucleosomes of our gene. As soon as the plants began to see the cold, that switched to red. And all the way through the winter, it was progressively changing to red. Then we brought the plants out in the spring. So then we measured what the analysis, the kind of modification. So now, what's the modification going to be in the spring? Is it going to be green or is it going to be red? So hands up for green. Hands up for red. Oh, yay. It's red. The gene is stably silenced because that's the silencing mark. It's inherited, even though the plants have come back into the warm, it's epigenetically inherited that off state. Now, the other thing, so we've talked about the epigenetic inheritance, why if you take a plant having had 3 or 4 weeks of cold, it stays off. But this other property that I introduced, which was unusual, was this quantitative silencing, why it was so slow. It wasn't that it sort of felt cold and switched it off in a sort of rather rapid way. And there are many temperature responses which are fast, but this is not. And of course, it needs to be slow because the plant needs to know it's had the whole of winter rather than a bad week in November and sort of make a terrible mistake and flower in December. So it's evolved to be very, very slow to make sure that the whole of winter is registered. So how can it be quantitative? Now, this is when I started working about 8 years ago with a quantitative biologist who trained as a theoretical physicist, and he looked at this and he said, but you've said to me that if you take the plants out at 2 weeks, they can remember an intermediate level of silencing. And you've also said to me that it's either green or red. So if it's green or red, how can you have a half level of silencing? I bet you any money this is not a nice gradual change as it looks like on the picture here, but it's a switch. They're green or they're red, and it's a population average. But of course, as you do molecular biology experiments, you grind everything up and you're averaging large numbers of cells. You then see this nice quantitative change. And he was absolutely right. Now, I put this in not to scare you, but just to introduce you to the fact we got into computational modeling of these epigenetic processes, which has actually become very important now because, as I said, this is important in cancer and lots of development. We need to be able to model switches in other systems because, of course, most other people are measuring quantitative changes. And actually what they're looking at is a switch. So they feed back on themselves. I've just put this in as a little taster. They're distinct states which reinforce themselves and oppose the opposing state. And that gives you a nice bistable digital state. So what happens is that each cell makes a decision. Shall I switch my FLC gene off? So now I haven't drawn a plant here, I've drawn a box, and each box, little box, is a cell. But if the gene is on, we've color-coded it blue, and if the gene is switched off in that cell, it's now white. And what we see is that each cell makes that decision to switch FLC off independently. And so slowly, 2 weeks, 4 weeks, 6 weeks of cold exposure, more and more cells have made that switch. But of course, when you grind these up, and you look on the molecular assay, it looks beautifully quantitative. So that's been a very big step. This we discovered in vernalisation, in FLC silencing, and now it's something everyone's looking at in the human system. Okay, so we've done memory, but I haven't told you anything yet about actually how the temperature is perceived. So let me just turn a little bit to the fluctuating temperatures and temperature. You know, it's a question I was asked a lot. So what is it that's the temperature responsive bit? What's feeling the cold? Is the thermometer somehow? And of course, when we did our experiments in the lab, we would put them in a nice constant cold room for 4 or 6 weeks, which is very atypical of what a plant normally sees. And this is actually what a plant sees. This is a field in Sweden where there's a temperature monitor at the level of the plant. And I was absolutely amazed at the level of variation a plant has to read. Every day, because it's a very, the sun's quite low in September and November in Sweden, they actually get the angle, heats the plants up quite high. I mean, you see up to 50 degrees. Look at the axis on the right, on the left here. You see that the plants are exposed to very high temperatures and then very low temperatures. And there's more fluctuation on a day-to-day basis than there is even on a week-to-week basis. So the plants have got to read fluctuating temperature. Now, there's clearly a period here in November, January, March where it was snowed on the ground, and that's why actually it's not fluctuating so much. Now, the one temperature from all our genetics, the one temperature monitor we had was a protein with a trendy little name called VIN3, vernalisation insensitive 3. Now, I'm not going to go into it very much, but interestingly, that accumulated slowly. So that fitted with this low probability switching that happened independently in every cell. So we thought, oh, well, that's it. We've just got a thermometer, and it's called VIN3. But no, actually, it's much more complicated than that. And again, this isn't really meant to scare you, but it's just as a reflection that when we've looked, every single step we've looked at in the process was temperature sensitive. VIN3 for sure, different aspects of VIN3 were temperature sensitive, straight on to FLC was also temperature sensitive, and the plant is monitoring lots of different aspects to actually then integrate those fluctuating temperatures. Such that, and I've color-coded them here, red and purple, that's all you really need to know. But what we then see is, if you then take that fluctuating temperature, what happens is that the plants register very low nighttime temperature through one mechanism. They register something about 15 degrees and below, not really cold, it just has to be below 15 to switch on a different, and this is actually the epigenetic silencing part. But if the temperatures go above 15, then something kicks in to turn over all of that accumulated memory of cold, because it actually switches off VIN3. So the plant is monitoring afternoon temperature peaks and evening or night troughs to work out what's happening and is winter progressing. And this is really a very different idea. People for a long time have thought they were averaging temperature through some biochemical molecule. So this is some relatively recent work to say, no, actually the plant's much more sophisticated than that. It's measuring peaks and troughs. Okay, now I'm just going to turn to the third part of my talk, and that's this adaptation to different winters. Now we had the molecular mechanism, we could then turn to say, well, how has that changed as plants have adapted to different winters? Unfortunately, Rubbertopsis grows all over the world, right up to the Arctic Circle, down to the Cape Verde Islands, and there's a big collection of natural variants where people have gone, collected them, and had them in a stock center, so we had them all. The strategy for the north is a winter annual strategy, so you'll see they all look, that none of these have been vernalized, they're all very late flowering. But actually the best, the most predominant strategy for ones from southern latitudes is this rapid cycling strategy. So lots of variation. Because we were interested in this winter period, we only picked the winter annual types. And we went in, and here I show a schematic of just two, one from very, very north Sweden, a place called Lervik, on the Arctic Circle, and another one from Edinburgh. And what we found was actually they needed different weeks of cold to get the same effect. So what I've said so far is, give it weeks of cold, it will silence this gene. And that silencing is required to allow all of the various other activators to function and for the plants to flower. But actually, the type from Edinburgh only needed 4 weeks, whereas the one from northern Sweden needed 12 weeks. Well, that fits, winter's longer in northern Sweden, but what has changed to allow that adaptation to happen? When we then did experiments where we put in the growth rooms in Norwich, gave them 4 weeks, this one from Edinburgh was fully satisfied for the vernalisation, but actually the one from the northern Sweden, the gene switched back on again if it hadn't had long enough cold. But there was nothing actually wrong with the vernalisation apparatus, because if you gave it 12 weeks, then the gene silenced. And so our molecular analysis so far says that what is happening is very similar to actually what we saw before. We have the gene on in the autumn, the gene goes off in the winter, we have the red modifications. In spring, they stay red, remember, just have the gene silenced for flowering. But if you haven't given this particular type enough cold, it switches back to green. And I put this in because actually when we discover what the differences are, there's just 4 changes in that gene that have allowed that to happen. So if you're thinking and worrying about changing winters and how fast things can change, 4 small changes make the difference between needing 4 weeks of cold and needing 12 weeks of cold. So it's remarkably facile to actually have that adaptive step. Now, we've gone on, and this is now very current work, to say, well, so there are the different types, and we know there are different abilities to, or the different factors that are important for perceiving the cold. But can we really understand the full extent of the natural variation and how it's operating in the field? So we've gone back and used Sweden as our sort of experimental area. And every green dot that you see on this Google map is a place in Sweden where we've found a natural population of a Rubbertopsis thaliana. And right at the top, you might be able to just see the Lervik is actually at the Arctic Circle. And so what we've done over the last 4 or 5 years is we've planted lots of Rubbertopsis in fields in the north and the south of Sweden. So we can put lots of different types that were normally from there, but then also types that weren't normally from there and were perhaps adapted to somewhere else, and say, how maladapted are they when we change the position? Now, that's meant that I've been able to do lots of lovely trips to northern Sweden. And in fact, this is one picture. It's a favorite picture of mine. It is a beautiful place. This is the World Heritage Site right on the Arctic Circle, a place called Lervik. And there's a picture of me and my collaborator who works up there at the University of Mid-Sweden, just to prove I was there. And somehow, every time I go, it's sunny like this. It's a very interesting place in that the ground is still going up, because the ice having left in the last ice age, the ground still is rising. And so there are little islands appearing all the time in the water, and lots of these are Rubbertopsis plants. Now, I get to go in the summer, but we've done these experiments where we've put out lots of Rubbertopsis, we sowed them in September, and then we've had students go every 2 weeks to these sites and harvest Rubbertopsis leaves and send them back to Norwich so we could do the molecular analysis. So I get to go in this, and the poor students are digging through, 3 feet of snow trying to find the Rubbertopsis plants, because, there's got a lot of snow. And the picture on the bottom is what they look like. They're very healthy, very happy under all this snow. And of course, they harvest the material and send it back, and then we can do all of the analysis. So this has been a very interesting, it's ongoing. We're getting a lot of information, and what's really surprising is how small a number of changes are required to have a very, very different requirement for the cold and different response to the cold. And what we've got to really understand now is the integration of all the temperature with these molecular switches to then understand from a plant point of view, from a crop point point of view, what we need to do to, if you like, climate-proof our crops for changing winters. And we really have to worry about this from a flowering point of view because, you know, there's a lot of global warming, clearly, for whatever reason. The temperature predictions are fairly dire as we go forward. But the photoperiod is not changing. So plants that have exquisitely evolved to certain temperature regimes and certain day length regimes are getting very confused because those are now pulling apart from each other. And we're having clearly already significant disruption for a lot of different crops, a lot of ecosystems because of this changing photoperiod temperature relationship. And a colleague of mine at John Innes, Judith Irwin, has been working very hard on the horticultural brassicas in terms of understanding and protecting our brassica horticultural cauliflower broccoli supply for the future. And she gave me this slide. Now, Rubbertopsis is a very close relative of the brassicas, so everything we learn in Rubbertopsis is immediately translatable to all of these vegetable crops. They're just more genes because it's a bigger genome, more FLC genes. And this was a picture she took just to show how different winters can be. So the winter of 2010, you might remember, was a very, very cold winter. The one where they had the satellite pictures where the whole country was covered in snow. There was one. And you can see this is the cumulative number of chilling hours between July and December in that year. You can see very little warm, a lot of chilling hours. The color coding is red and blue is more. So more chilling in 2010. The exact same period the year later, completely different. So our plants and our crops have got to have that flexibility to cope with, you know, these very different changes in winter. And yet we want them, or the supermarkets all want them scheduling to come out exactly at the right time for when they want the production and the supply. And certainly that year, the 2010 year, there was really a big problem in the cauliflower and the broccoli industry. Very short supplies because of the bad weather. Okay, so I've taken you on quite a long journey from starting from a student doing experiments on chloroplasts, getting excited about flowering, deciding to work on flowering as a new investigator, and then the journey through really opening up the genetic basis of flowering and then the molecular analysis of vernalisation to now the current work on adaptation. And I hope that some of you will have taken out different bits of the seminar, but there are three sort of take-home messages I just want to reiterate as ones to sort of try and take away with you. And the first is this genetic simplicity of what was thought for 50 years, this massive physiological complexity in flowering time. You know, there were communities of researchers working on all sorts of different plants and crops, absolutely sure that photoperiod would be regulated in an entirely different way to vernalisation, to hormones, and they would all have to be sort of studied separately. But in fact, through the fairly straightforward molecular mutation analysis in this reference plant, which is actually a weed, but it's a very useful weed, and it's got the same set of genes as every other plant, we could come up with this relatively straightforward genetic regulatory hierarchy, which then has helped us go in and understand flowering in all plants. And I think once you get to sort of a simplicity, it probably means we get into an understanding. The second is that the plant biology field was completely transformed by the adoption of this reference organism, Rubbertopsis thaliana. So whoever's, you know, it's like pass the parcel, who's ever got it as the music stops, those plants. Take them home, stick them in the garden and watch them. They'll proliferate, they'll drop all of their seeds next year and you can have a nice little experiment ongoing. But it's been a huge boon to plant biology. We really have been able to dissect all sorts of things, disease resistance, growth stature, habit, all sorts of things that have been very important for, and they will be for, biotechnology. And also is showing that 50% of a plant genes are exactly the same as in humans. You can take the plant gene and put it in a human and it will work just fine. So we learn a lot from plant biology because it's easier to do plant biology to make mutants to clone genes that can be applied directly to the human system. And I also described how theoretical modeling was really a fantastic thing to add to the molecular basis to actually get detailed mechanism. And then the third and the last message is really this epigenetics, that we've got, in this case, an epigenetic modification of the proteins. We just call them green and red. They're chemical modifications of these proteins that make up these nucleosomes. They are induced by the cold, but then even when the cold goes away, they are epigenetically stable and inherited from daughter to granddaughter of the cells. But they're reset every generation, erased in the seed, so that every generation of seedlings now needs to be revernalized. But our detailed analysis of this mechanism now is actually being used in lots of systems and lots of animal systems as well, because it's much easier to do the mechanistic dissection of epigenetic switching in this system than it is in animals. And last but not least, I want to end with a picture of the people who've been involved in the work. You know, there were many people over many years, and this is now the current group. We're all at the John Innes Centre in Norwich. I showed you a picture of that early. The team labelled in red are all the theoretical modelers. Martin Howard is the computational biologist I told you about, the theoretical physicist in the middle. And his team, Rhea and Cecilia, they are mathematicians and physicists. But we sort of work as one jumbo group now. And the ones in white are the people in my group. Some have left if their names aren't there. People have come, thank you. Some of them come to support me tonight from my team. And what's a joy is to have a huge range of nationalities. We get funding from the ERC, or the European Research Council, from a lot of different, from BBSRC, which is the UK Council, Marie Curie, which funds people from all over the world to come to the UK. We have a huge number of different nationalities that all work together very effectively, and then leave and set up labs, and then end up still collaborating with us. So we've sort of got a rather large Dean family over the world working on things related to vernalisation and epigenetics. So with that, I will thank you very much, and I will happily take some questions.